Well, hello and welcome back to Science in the Age of COVID-19. Today we are super thrilled to welcome back Jesse Bloom. Jesse first joined us, I believe, in the fall. Jesse is an investigator of Howard Hughes Medical Institute and professor at the Fred Hutch in Seattle. Last time Jesse was here, he presented us an amazing study on mapping all mutations to SARS-CoV-2 spike protein and the effect on its primary receptor ACE2. Jesse's data set was super rich and revealed such things as E484K being a potential mutation of concern before it was seen in the wild. So very useful studies to map out what this virus has access to and how that will be able to hurt us. He has now updated that study, mapping not only binding to the receptor, but also escape from neutralizing antibodies. So really timely in how this will affect the monoclonal antibody treatments and, of course, what everyone is worried about, vaccine protection. And lastly, Jesse is super committed to open science, and he has made his slides freely available in addition to the recording. So Jesse, blow us away.
Okay, hello everyone. So hopefully you can hear me talking. So my name is Jesse, and I'm going to talk to you today about interpreting the evolution of SARS coronavirus 2. And the slides I'm presenting today are available at this link, and I just posted them in the chat. So I'd like to start by taking a step back. I'm going to be talking about the evolution of this particular virus today, but it's really useful to think about how people have historically studied evolution. So for most of the history of evolutionary biology, people didn't know about genes or genomes or DNA or RNA or anything like that. So for instance, here's Ernst Haeckel's Pedigree of Man, and you can see that he's arranged all these different species in a tree-like structure based just sort of on looking at their similarity in gross phenotype. So it was only about 55 years ago that it became possible to also start to study evolution in terms of changes in genotype. So one of the first studies of this kind was by Emil Zuckerkandl and the famous chemist Linus Pauling. And what they did is they obtained the myoglobin protein sequences from a variety of species, and they arranged these sequences in sort of a tree-like structure based on the similarity of the sequences. And we've all become very used to thinking about evolution in this way. But at the time, if you go back and read their paper, which was actually the proceedings in a conference, this approach was kind of heavily criticized by famous evolutionary biologists like Ernst Mayr or George Gaylord Simpson as sort of being very trivial, because what could you expect to really learn about the interesting features of these species just based on looking at mutations in some protein? And in fact, Zuckerkandl and Pauling noted that fact, and they discussed how changes in genotype affect phenotype is really complex. So they said there's no reason to expect that the extent of functional change in a polypeptide chain is proportional to the number of amino acid substitutions. They said it's the type rather than the number of substitutions that is decisive. So in the 55 years since that study was done, we've now gotten super good at characterizing evolution in terms of genotype. And we've gotten, in fact, so good at doing this that most people have forgotten that evolution even ever used to be studied any other way. So for instance, here's, you can go to GISAID and you can download over almost 600,000 SARS-CoV-2 sequences, and you can build these super high resolution phylogenetic trees, such as the one shown here, which tell you how all these viruses are related at the level of genotype. But we still often want to know how these changes in genotype are also affecting phenotype. So here's an article on this topic. It says the coronavirus is mutating. What does that mean for us? And the point I just want to make here is that this question that everybody's so interested in right now, what do these mutations mean for us, is really a special case of this more general question that's been studied for a long time now in terms of proteins, for instance, which is how does sort of the number of amino acid substitutions, which is easy to observe, relate to the type of functional change that might occur. And so in my lab, we're really interested in studying this relationship of how changes in genotype affect phenotype in the context of viral proteins. So we're interested in the general problem where you might have a viral protein and you might want to make some changes to this genotype of this protein. So for instance, amino acid substitutions. And then you might want to know how those substitutions affect the biochemical phenotypes of that protein that we think might be relevant for its function or for fitness in nature. So in this talk, I'm going to discuss sort of looking at this general problem in the case of a specific viral protein of a lot of interest. So first, I'm going to give some background on that protein, which is the SARS-CoV-2 spike and its receptor binding domain. Then I'm going to discuss some studies of historical coronavirus evolution that give us some sense of how we might expect these viruses spikes to evolve in the level of phenotype. Then I'm going to really zoom in and discuss understanding how mutations affect the function of the SARS coronavirus 2 receptor binding domain. And then finally, the thing that I think most people are really interested in right now, which is how mutations will affect recognition of this viral protein by both monoclonal antibodies and the type of polyclonal antibodies that, for instance, might be elicited by infection. So first, just a little bit of background on the protein that I'm discussing. So on the surface of SARS-CoV-2 virions are this protein called spike, which is shown here in red. And the function of this protein is to get the virus into cells. So one important part of the spike is the receptor binding domain, shown here in purple. And so this is an autonomously folding subdomain of the spike. And the function of the receptor binding domain is to bind with high affinity to a protein called ACE2, which is expressed on the surface of some human cells. And this binding of the receptor binding domain to ACE2 is the first step in the initiation of cellular infection. We also now know that neutralizing antibodies, so antibodies that are able to directly block viral infection, target the viral spike. And they often bind to the RBD, and in many cases simply function by sterically interfering with the binding of the RBD to ACE2. And so this turns out to be a super important class of antibodies, and I'll get into some specific numbers later in the talk about how important this specific type of antibody is for immunity to this virus. Okay, so that's some background on the SARS-CoV-2 spike and its receptor binding domain. So before I get into SARS-CoV-2 in detail, I now want to sort of take a step back and broach this general question of how might we expect the spike of this virus to evolve in terms of the phenotypes we care about. And so this is a really important question because only sometimes does virus evolution at the level of genotype lead to changes in antigenic phenotype. So here I'm giving two classic examples of viruses that are opposite extremes of this spectrum. So these are both respiratory RNA viruses. So they're both viruses that transmit by the airborne route and have very high mutation rates. One of them is measles virus. That virus does not evolve to escape immunity. So in the old days, people were infected with measles virus at most once in their lives. And then a vaccine was developed in the 1960s and it still works great today. At the other extreme is influenza virus. So this virus evolves rapidly to escape antibody recognition. As a result, people are infected with the virus about every 5 years, and the vaccine needs to be updated annually to keep pace with that evolution. So you naturally might want to ask, where might a coronavirus fall in this spectrum? So early in this pandemic, you often read things about how coronaviruses were evolutionarily stable or didn't evolve antigenically. So here's an article on that topic from March of 2020. And the reasoning that was sort of proffered to support this idea is that coronaviruses are the only RNA viruses with a proofreading polymerase, meaning that they have a mutation rate that is somewhat lower than the mutation rate of something like influenza virus or measles virus. However, we were not impressed by the evolutionary depth of that argument, and so decided to study mutations to these viruses anyway. And the reason we thought that wasn't a very good argument is it's important to understand that evolution isn't the same as mutation. Evolution in phenotype involves three related factors, which are the underlying rate at which mutations arise, the phenotypic impact of those mutations after they've appeared, and then how selection and also drift act on those mutations to cause them to disappear or spread in a population. And so these are really complicated, and it's hard to sort of forecast them for a virus. So the approach we took was to not try to really predict what's going to happen with SARS-CoV-2 at the level of will it undergo antigenic evolution, but to just look at the past behavior. So there actually isn't any direct past behavior of SARS coronavirus 2 because it's only been known to infect humans for a little over a year, but we can look to its relatives. So it turns out that there's a variety of other coronaviruses that circulate in humans and cause common colds. So about 30 or 35% of all common colds are caused by these coronaviruses. And one of these is called coronavirus 229E. So this is a coronavirus that's been circulating in humans since at least the 1960s and probably much longer. And many people have never even heard of this virus because it does not cause particularly serious disease. So what we did is we went to the available sequences in GenBank and we just reconstructed a phylogenetic tree of the coronavirus 229E spike protein going back to the oldest non-lab adapted sequences we could find, which were in the very late 1970s. So here are sequences from the late 70s and early 1980s, and then down here are sequences from the last few years. And so I hope you can appreciate a couple of things about this phylogenetic tree. So first of all, this phylogenetic tree has what is called a ladder-like shape. So there's a major trunk lineage that I'm tracing with my mouse here, and then all of the branches are sort of short, stubby branches that come off of that trunk. And what that pattern means is that although the virus is changing over time, the standing diversity at any given time is low, because as the trunk lineage moves forward, the other lineages die out. So this is a pattern that's seen for some viruses, such as influenza virus. It's not seen for other viruses, such as HIV. So for HIV, the tree would have lots of different long branches that all coexisted because there's high standing diversity in HIV as well as change over time. A second, so this sort of phylogenetic tree basically means that there's one lineage that's constantly winning out and displacing the other lineages. Another thing I hope you can appreciate is I've colored the tips of this tree by country. And admittedly, in a lot of time frames, only one country was sequencing this virus because it's not heavily studied. But in any time frame where multiple countries were sequencing the virus, such as Japan and Australia here, or Germany and the US here, you can see that the sequences cluster by date rather than by country. So this is, again, a characteristic we often see of rapidly transmitting respiratory viruses, which is that there's very little persistent geographical structure. So the world is, approximately a well-mixed flask for these viruses because they transmit very rapidly across the human population. So one feature of a phylogenetic tree that has this basic shape is that actually just time of virus isolation is a pretty good proxy for where you are in this sort of evolutionary trajectory. So we just experimentally generated spikes at 8-year intervals. So here in 1984, here in 1992, 2001, 2008, and 2016. And we generated these spikes in the lab. And then what we did, so we can obviously see from the tree that they're changing in sequence, but we wanted to know, is that changing phenotype? So then what we did is we took each of these spikes and we asked how well was infection by a virus with this spike neutralized, so blocked by the antibodies in human serum. And so here I'm showing this result for a single human serum sample collected in 1985 from someone who was 26 years old at the time. So that the portion of the plotted time frame during which this individual is alive prior to the serum collection is in blue. And the y-axis here is how well that serum neutralizes the virus. So a higher number means it neutralizes the virus better. And you can see that the serum neutralizes the contemporaneous virus quite well, but as we look to sort of future viruses, the neutralization titer goes down. Again, this is all the same human serum sample. So this suggests that as this virus is changing through time, those mutations are eroding the ability of this person's serum to neutralize the virus. And in fact, in general, if we look across additional human serum samples, we see that the antibody immunity generally will recognize viral strains that circulated during that person's life, but not strains from far in the future or far in the past. So here's the example I just showed you. Here's some serum collected in 1995. Again, person was alive during the blue shaded period. You can see they neutralized the older viruses, but not sort of future viruses. Here's serum from an adult collected recently. It neutralizes the whole span of historical viruses. Here's serum from a child collected recently. It only neutralizes newer viruses. So we think this pattern arises from the fact that coronavirus 229E, like most common cold coronaviruses, infects people every, probably 3 to 5 years. And it's probably being able to reinfect people because the virus is evolving to escape from their prior immunity. And then once it reinfects them, they have immunity to that new virus, but they're not going to have immunity to sort of viruses that will evolve far in the future. So this is showing that coronavirus 229E is evolving in terms of antigenic phenotype much more similarly to influenza virus than to measles virus. However, one other interesting thing that we noted is that this viral evolution erodes the antibody immunity of different people at different rates. So for instance, if you look at the far left and the far right of this plot, here are two different people. Both of them have similar neutralization titers, a little bit greater than 1 to 100 against the 1984 virus. Yet this person's antibody immunity was essentially completely eroded, at least in terms of its neutralizing activity, by 8 years of viral evolution. Whereas this person's immunity, although it's gradually eroded by viral evolution, it stands up to it very well. So even 3 decades later, they can still neutralize the descendant viruses from the one in 1984. And we expect that these differences are because different people are making antibodies that are binding to slightly different parts of the viral spike. And we're right now trying to figure out what is sort of those differences in specificity between evolution-resistant sera, like this at the right, and evolution-sensitive sera, like this at the left. Because you can imagine if you were to try to make a vaccine against a coronavirus, you would like it to elicit evolution-resistant immunity, such as that at the right. And then one other point that I think is going to be relevant for those of you who are sort of SARS-CoV-2 aficionados is that although the coronavirus 229E spike has a different receptor and it is fairly diverged from the SARS-CoV-2 spike, it does have the same domain and subdomain structure. In particular, there's a receptor binding domain that uses some loops to bind to the receptor, which is aminopeptidase N, and then there's also an N-terminal domain. And if you plot over this 40-year time frame I was talking about where the sequence variability is in this spike, you can see the sequence variability is especially concentrated in the binding loops in the receptor binding domain. And then there's also quite a bit of sequence variability in the N-terminal domain. And for those of you who follow SARS-CoV-2 evolution closely, you'll know that in these new emerging lineages we're seeing in SARS-CoV-2, there's a lot of mutations in the receptor binding loops, such as N501Y or E484K or K417N. And then there's also some mutations in the N-terminal domain. So it sort of seems like what's happened in the past with coronavirus 229E might be similar to what's happening now with SARS-CoV-2. Okay, so now I've given you some background on the SARS-CoV-2 spike and its receptor binding domain, and I've also told you that coronavirus spikes, at least coronavirus 229E spikes, can undergo genetic evolution that leads to change in their antigenic phenotype. So now I want to talk about really trying to zoom in and specifically understand how mutations affect the function of the SARS-CoV-2 receptor binding domain. So what we'd really like to do is prospectively map how all of the mutations to this protein affect its phenotype so that we can sort of interpret and perhaps to some degree forecast how the virus might evolve. So unfortunately, it's impossible to measure true viral fitness in the lab. Fitness is a property of the virus's ability to transmit in the human population, which cannot be fully modeled in any sort of lab experiment. So we instead decided to focus on 3 biochemical phenotypes, which we can measure and which we think crucially contribute to this true viral fitness. And those biochemical phenotypes are does this protein fold properly? Can it bind to ACE2 with high affinity? And then can it be recognized by antiviral antibodies? And at sort of a cartoon level, we imagine the evolutionary pressure can about be approximated like this. As the virus evolves, there's going to be pressure to stay good at having this protein fold properly and bind to ACE2. So there's going to be purifying selection to stay good at these two phenotypes. And then at the same time, the virus is going to be under diversifying selection to change its ability to be recognized by antiviral antibodies as much as possible, because that would allow it to infect people who had, antibodies from prior infections. So first, I'm going to talk about looking at these first two phenotypes. So in order to measure these biochemical phenotypes, we just used a yeast display system. So we took advantage of the fact that the RBD is an autonomously folding subdomain of spike, and so it can be expressed on the surface of yeast. We can then label the RBD on the yeast surface using an epitope tag, which we can detect with a fluorescent antibody. And it's generally thought that if the protein isn't properly folded, it generally isn't displayed on the surface of the yeast with high efficiency. And then we can also incubate these yeasts with something that binds or is bound by the RBD, such as in this case, fluorescent ACE2. So I think you can imagine that, for instance, if you had these two labels, you could then very easily use flow cytometry to separate their yeast by both how much RBD they have on their surface and how well that RBD is binding to ACE2. And so this basically allows us to use a flow cytometer to measure these biochemical phenotypes. And then the important thing is we don't just have yeast expressing sort of the wild type or original RBD sequence on their surface. We actually have a library of yeast, each of which are expressing a different RBD mutant. And then those yeast have a 16 nucleotide barcode, which we can use to determine which RBD mutant they have on their surface. And that means we can just take the yeast and sort of deep sequence for this short 16 nucleotide barcode and figure out which RBD mutants we have after we do flow cytometry. And so we actually have lots of RBD variants in these libraries. We have almost all of the single amino acid mutations, and we also have many double and triple mutants in these libraries. For the purposes of the talk today, I'm just going to be talking about the effects of the individual mutations, but certainly you can imagine that looking at epistatic interactions among multiple mutations in the RBD is also becoming increasingly important as more and more mutations accumulate. Okay, so just by using this combination of deep sequencing and yeast display, we can build phenotypic maps of how mutations affect these two phenotypes of folding and ACE2 affinity. So here's just an example of what those maps look like. So these are two heat maps. The top one represents the biochemical phenotype of ACE2 binding. And in particular, it's the log 10 apparent KD of the receptor binding domain for ACE2. And then the bottom represents the expression of properly folded RBD on the surface of the yeast. And then each column here is a different mutation. And the mutations are a different site, and each row is a different mutation or different amino acid. So each cell is a mutation. And they're colored red like this if the mutation is bad for that biochemical phenotype, white like this if it has no real effect, and blue if it actually enhances that biochemical phenotype. So you can observe a couple of things if you just kind of look through these heat maps. There's a lot of red because it's well known that most mutations to proteins are deleterious. There's quite a bit of white. It's also well known that typically there's a lot of fairly neutral mutations to proteins. And then there's some scattered blue, which means there's some mutations that enhance these biochemical phenotypes. And so these maps are kind of boring if you don't care about the receptor binding domain. But right now, a lot of people care about the receptor binding domain. So you can go look up specific mutations that are observed and try to get a sense of what they do. So for instance, one mutation that's become very famous is called N501Y, which is right here. And this is a mutation that's found in a large number of the new emerging viral lineages. And you can look at our map and you can see that that particular mutation enhances the affinity of the receptor binding domain for ACE2, which could be part of the reason it's been favored by selection. You can also look at other famous mutations. So E484K is another famous mutation, probably more for its effect on antibody recognition. But you can see that mutation also very slightly enhances the affinity for ACE2. Another famous mutation, which is in like the B.1.351 lineage, is K417N. And that mutation is right here. And you can see that mutation actually somewhat decreases the affinity for ACE2. And in fact, that mutation's in general only been observed in conjunction with other mutations like N501Y. So based on these phenotypic maps, we can speculate that maybe this affinity decreasing mutation needs to be compensated by an affinity enhancing mutation. And in general, we hope these maps are useful for when people hear or observe new mutations in the receptor binding domain, they can go look up and see how those affect these two key biochemical phenotypes. And you can also display these in various other ways. You can project these phenotypic maps onto the structure of the RBD. Here's a link to do that. And they've been integrated into various browsers, such as the UCSC SARS-CoV-2 genome browser, where you can look at these biochemical phenotypes in conjunction with other information related to, let's say, the virus's evolution in nature. Okay, but rather than dwell on those maps more, what I want to sort of get to is how we can now use those and the experimental platform I discussed to get at the question, which I think is of the most intense interest in the study of SARS-CoV-2 right now, which is how might mutations affect antibody recognition of the receptor binding domain. So I don't think it takes a ton of imagination to imagine how if you had an antibody that binds to this viral protein, you could use these yeast to find the mutations that reduce that binding. So we just take the yeast, we incubate them with the antibody, and then we run them through a flow cytometer to identify those mutants that bind to less antibody than sort of most of the rest of the yeast. And we do some experimental and computational filtering to get rid of yeast that just have no RBD or misfolded RBD. And then we just deep sequence what were all the RBD mutants we started with and what are the ones that could escape the antibody binding. And I'm going to represent that phenotypic data in the following format, where for each mutation, I draw a tall letter if that mutation strongly escapes antibody binding. In particular, the height of the letter is proportional to the escape fraction, which is the fraction of all the yeast with that mutation that fall in the antibody escape backspin. And so when I show these maps of how mutations affect antibody binding, what you need to remember is if there's a tall letter, that indicates that that mutation strongly escapes antibody binding. So, okay, what do these escape data actually look like in real life? So here's an escape map for one antibody called CoV2 2499. This was an antibody isolated by the Crow lab at Vanderbilt from a SARS-CoV-2 convalescent individual. And we measure the effects of mutations throughout the receptor binding domain on antibody binding. Here I'm just zooming in on some key sites. And all of the sites that I'm not showing have look like this. They have no mutations that escape antibody binding. So you can see there's a few sites like 443, 445, 446, 447, and down here where mutations strongly reduce antibody binding. And in fact, we can understand that in terms of the structure of the molecule. So here is, sorry, one second. Here's let me see if I get the colors to load here correctly. Okay, so here's the structure of the receptor binding domain in this light gray color bound to ACE2 in this sort of darker color here. And then here is every site in the receptor binding domain, how much mutations at that site affect antibody binding for this particular antibody. And you see that most of the sites mutations have no effect, but there's a few sites like 447, 446, 496, et cetera, where mutations strongly affect antibody binding. So if we look to see where those sites are, they all are in one region of the receptor binding domain, which is presumably where the antibody binds. And so this has sort of told us both the physical footprint of the antibody with the receptor binding domain and what are the mutations that disrupt that interaction. And just a point I want to allude to is that you can imagine, why do we need to do this experiment? Could we have figured this out by crystallizing the antibody with the receptor binding domain? And in fact, we could have gotten complementary information or similar information doing that. But as I'll allude to later on, these experiments that I'm describing can also be done with polyclonal mixes of antibodies, which are less amenable to structural biology needs. Okay, so here is this, again, map of how mutations are affecting this biochemical phenotype. So one question we wanted to ask is, just by measuring these effects of these mutations on these biochemical phenotypes, can we actually explain how the virus evolves under antibody pressure? So to do that, we worked with our collaborators to grow a VSV virus, which is sort of a biosafety level 2 surrogate for a SARS-CoV-2 spike-expressing virus in sort of very high throughput in the presence of antibodies. So for instance, if we grow this virus 16 times in the presence of this antibody CoV-2 2499, we get several different outcomes. So 11 of those 16 times, the antibody controls the viral replication and there's no detectable viral replication. But 5 of those 16 times, the virus manages to end up growing in the presence of the antibody. And each of those 5 times, it turns out the virus has acquired a mutation. The mutation is G446D 3 times and Q498R 2 times. So we now wanted to ask, all right, we've made all these biochemical measurements. Can we understand why these 2 particular mutations emerged? So first, we just went and looked at this first biochemical phenotype, which is how do the mutations affect antibody binding? And so here I'm showing all of the sites with tall letters. And you can see that, in fact, the 2 observed mutations, G446D and Q498R, are among this set of tall letter mutations. So they do reduce antibody binding. But looking at this biochemical phenotype alone has low predictive power because there's a lot of other mutations that also escape antibody binding that did not appear in the context of virus. But we have some additional information about how this evolution is constrained. So first of all, here I'm showing all amino acid mutations. If we just zoom in on those mutations that can be accessed by a single nucleotide change to a codon in the virus, there are now fewer single nucleotide accessible binding escape mutations, and the 2 that are observed in the context of the virus are among them. So the predictive power has gotten better, but it's still not that great. However, here I'm just showing one biochemical phenotype, how mutations affect antibody binding, but we know that the virus is also under pressure to continue to be able to infect cells. And we've in fact measured how mutations affect phenotypes relevant to that, such as affinity for ACE2. So now I've colored these letters by how good the affinity of that particular mutant is for ACE2. So the letters are colored brown if that mutation has high ACE2 affinity, and yellow if it has poor ACE2 affinity. So you can now see there are only actually a few mutations that are both tall letters, so escape antibody binding, and brown, so still bind to ACE2 well. And the actual mutations that arise in the context of the virus are among them. So we're getting like decent predictive power here. We've narrowed down to a small set of mutations, and the ones that actually occur are among that set. So a more compact way to plot everything that I just described to you is as follows. So here's a scatter plot. Each point on this scatter plot is one of the about 4,000 amino acid mutations you can make to the receptor binding domain. And then the x-axis indicates how strongly that mutation disrupts antibody binding. And the y-axis indicates the affinity of that mutant for ACE2. So teleologically speaking, which I know you're never supposed to do in evolutionary biology, the virus sort of wants to escape by getting mutations here in the upper right, because these are the mutations that reduce antibody binding while also retaining good ACE2 affinity. And after we gray out with these X's, all of the mutations that require more than one nucleotide change to the codon, you can see that there are only a small number of mutations that satisfy sort of these 2 biochemical constraints. And the ones that actually arise in the context of the virus, shown here in red, are a small subset. And in fact, we've done similar experiments for a number of other antibodies. And what we consistently see is that when the virus is grown in the presence of the antibody, it escapes via mutations. In every case, the observed escape mutations are shown in red, that are basically as far in the upper right as it's possible to get, given the particular biochemical constraints on the phenotypes of these mutants. So I think this is cool that just by looking at these biochemical phenotypes, we can actually gain a lot of explanatory power to understand how the virus evolves under a particular selective pressure. And so we've now built these escape maps for many of the lead clinical antibodies that are being used as antibody therapeutics. In particular, we've done it for the antibodies in the Regeneron cocktail, the antibodies being developed by Eli Lilly, and then the antibodies that are currently in advanced clinical trials by AstraZeneca. And if you're super interested in any of this, here's the links to the papers, and you can also go click on these and sort of look in great detail at, whoops, that was the wrong link there. Look in great detail at sort of exactly where the escape mutations for any given antibody fall. So for instance, for this Eli Lilly antibody, which is called bamlanivimab, you can see that the biggest escape mutation is site E484, and the other escape mutations are sort of in similar regions of the tertiary structure. And so using this is actually useful because people are, these antibodies are being used for treating people with, there's some evidence that they're helpful if they're given early to people with mild disease at the time. And we can now look at the emerging viral variants and see which of them have mutations that escape binding by these antibodies. And so, for instance, here are some of the most major viral lineages that you hear about in the news. And many of these viral lineages have this E484K mutation. And you can see that mutation, unfortunately, escapes from this bamlanivimab or LY-CoV555 antibody being developed by Eli Lilly. You can see that this viral lineage from Southern California, which has the L452R mutation, also escapes from that antibody. Although fortunately, some of the other antibodies in some cases are somewhat or much more resistant to the mutations that have been observed so far. And so because this virus is undergoing rapid evolution and because there's a lot of antibodies that are being developed for clinical use, we think having this sort of information could be informative to sort of decide which antibodies might be useful to sort of use in any given area, depending on which viral lineages are circulating there. Okay, so far I've told you about monoclonal antibodies, which I think everyone views as sort of a stopgap measure to treat infected people while people are getting infected and potentially seriously ill. And again, just to kind of make a obvious point, a feature of a monoclonal antibody is it's a defined antibody that's produced synthetically, typically, that binds to a specific location on the virus. And so typically, if the virus gets a mutation in that specific location where the antibody binds, the antibody doesn't bind anymore. However, the biggest hopes for controlling this pandemic rely on polyclonal antibody immunity that might be elicited by infection or vaccination. And so the human immune system can make a very, very large number of different antibodies in response to infection or vaccination. And those antibodies, at least in principle, can bind to many, many different epitopes on the virus. And so when this is the case, you might imagine that these sorts of polyclonal antibodies can be much more resistant to viral evolution, because even if a mutation knocks off a particular antibody, such as this purple one, there's still all these other antibodies left. Importantly, sort of the functional diversity of the polyclonal antibody response is in principle. So the immune response can target many different epitopes, but there's no fundamental law of immunology that says that a whole bunch of the antibodies can't end up binding to the same epitope. And in fact, we know that the extent of focusing of polyclonal antibodies is probably an important factor in shaping viral antigenic evolution. So for influenza virus, if you look at human polyclonal antibody immunity, it's often highly focused, such that it's not unusual for a single mutation to the virus's hemagglutinin protein, its main surface protein, to reduce neutralization by that polyclonal antibody immunity by greater than tenfold. In contrast, for measles virus, the polyclonal antibody response is much better balanced across many epitopes in the viral surface proteins with similar potency, so that typically no single mutation has a large effect on neutralization. And instead, you need many, many different simultaneous mutations knocking out many of these epitopes to get strong escape from neutralization. And also, although it's obviously impossible to sort of prove the causes of epidemiological phenomenon, I think these differences could be one important reason why influenza virus is so much more capable than measles virus of evolving to escape from antibody immunity. So we wanted to then study, what does this sort of polyclonal antibody immunity look like in the context of SARS-CoV-2? And so first, we just wanted to figure out which part of the spike protein to focus on. And so we did some experiments, which sort of repeated some earlier experiments by Piccoli et al., to show that most of the neutralizing activity of most convalescent human sera is from RBD binding antibodies. So this plot shows the results of serum from a whole bunch of different individuals that had been previously infected with SARS-CoV-2. And on the x-axis here is the neutralization titer. So a bigger number means they neutralize the virus better. So most people have neutralization titers between 1 in 100 and 1 in 10,000. And those neutralization titers are these closed circles here. And then what we did is we took these serum and we biochemically depleted only the antibodies that bound to the receptor binding domain, which recall is just one part of the spike, although the most key part for binding to receptor. And then we remeasured those neutralization titers. Those are the open circles. And so what you can see is that in almost every case, the neutralizing activity of the sera goes down dramatically when you get rid of the antibodies that bind to the RBD. So in this case, greater than 99% of the neutralizing activity is RBD directed. So this is not always the case. There are certain individuals like this one here, where the RBD binding antibodies only make up a small fraction or a modest fraction of the neutralizing activity. But in general, the RBD binding antibodies are the dominant contributor to the neutralizing activity of human sera. So this shows that at least the functional activity of that antibody response is at least focused on this one protein subdomain of the virus. We next used the same yeast display system I just described to you to then map how all mutations within the RBD affected binding by convalescent human sera. So I'm now again showing these logo plots where big letters mean that mutations at that site escape from antibody binding. And I'm coloring the letters based on sort of three major regions in the receptor binding domain where we tend to see these mutations. And here I'm sort of showing 4 representative-ish human sera. And if you want to see more, you can go look in the paper here. But I think this is enough to capture the main trends. And so, for instance, in the case of this individual, you can see that mutations at site E484 have a really big effect on that antibody binding. And in particular, we think the major epitopes for convalescent human sera on the receptor binding domain are this receptor binding ridge epitope, which prominently includes site E484, and also this sort of 443 to 450 loop, which is sort of on the other side of the receptor binding motif shown here in light blue, that's dominantly targeted by a smaller fraction of individuals, such as this one shown here. And sort of looking across a lot of sera, including more than the ones I'm showing here, something that really pops out is that the most important site in general for reducing binding of human polyclonal antibody immunity is site E484. And here you can see in many of these individuals, E484 is the most prominent or one of the most prominent sites. But as for coronavirus 229E, people are affected differently by different viral mutations. So this person's immunity is highly focused on E484. Mutations here have a huge effect. This person's immunity is more focused on this 443 to 450 loop. This person's antibodies seem to have very broadly distributed binding activity such that no mutations have a particularly large effect. And again, this could map back to the phenomenon that at least in the case of coronavirus 229E, we see that as the virus evolves, the phenotypic impacts for antibody recognition differ between the serum of different people. And importantly, these differences in sort of antibody binding that I'm showing you using this yeast display system tend to validate pretty well in neutralization assays. So here's just one example. Subject C is this individual that had the antibodies strongly targeting site 484. And you can see the neutralization titer goes from being around 1 in 10,000 if there's no mutations in the virus to more like 1 in 100, actually off the scale of our cutoff right here, if you put in this single E484K mutation. But for this other individual, subject G, the E484K mutation has no real effect on their neutralizing antibody recognition, but mutations at sites like 446 have a big effect, consistent with the differential targeting of their antibody immunity. And so one thing we can do is we can sort of integrate these data across many sera to assess which current mutations have the biggest antigenic effect. So here what I'm showing on the x-axis is the frequency of mutations at each site in the receptor binding domain among all sequenced SARS-CoV-2 isolates. And then on the y-axis, I'm showing you the total escape at that site averaged over all of the human sera or plasma we've looked at. And again, as I pointed out, this averaging isn't always what you want to do because there is real variation in the human population. So we don't want to mask that, but averaging gives a sense of what's going to impact sort of the most people the most. And so you can see the mutation to really watch out and be concerned about is clearly at site 484. So that mutation is fairly prevalent, and it also is clearly the site where mutations have the biggest effect. And in fact, this mutation is present in a number of these emerging viral lineages, such as the B.1.1.7 lineage, the B.1.351 lineage, the P1 and P2 lineages, and also this new, I think it's B1.526, although I have trouble with all this nomenclature, that was recently identified in New York. And so probably this mutation is being recurrently selected, at least in part because it's such a potent immune escape mutation. We can see that some of the other kind of prominent and common mutations, such as at site 501, have much less of an antigenic effect. And then we can see there are some mutations that are at lower frequency, but still non-negligible frequency that look like they'll also have pretty appreciable effects. Okay, so just to sort of conclude, relating sort of change in genotype to phenotype is this old challenge in evolutionary biology. So I told you how Zecker, Candle, and Pauling were worrying about this back 55 years ago, looking at these myoglobin sequences. And it remains a big challenge today, for instance, in the study of viruses like SARS-CoV-2. And in particular, I think that this is really increasingly becoming the bottleneck. So if you go back to Zecker, Candle, and Pauling, they spent a lot of time, and lots of people spent a lot of time getting those myoglobin sequences. Now we've gotten really good at obtaining genotypes. So sequencing viruses is something that's done a lot now. And the real challenge we face now is sort of interpreting what all those sequence data mean in terms of the phenotypes we care about. And so hopefully I've told you how we're trying to take some new approaches to map how mutations affect some of these key biochemical phenotypes in order to make it possible to better interpret the evolution we can now observe so readily at the genotypic scale. And then finally, I'd like to end with some thanks. In particular, I'd like to really call out Tyler Starr, who's a postdoc in my group who developed this whole yeast display system actually originally in the context of an entirely different project, and then adapted it to look at SARS-CoV-2 receptor binding domain over the last year. And Ali Greeny, who's a really great MD PhD student who's been working with Tyler on this and leading all of the sort of antibody and serum mapping part of it. Rachel did the work on coronavirus 229E I discussed. Sarah's a former grad student who built a lot of the tools for interactive visualization I showed. Kate has done a lot of our work on sort of viral neutralization assays, which I didn't discuss as much, but really is important for this work. And then a number of other members of my lab have contributed in important ways. I'd also like to thank our collaborators on various aspects of this work and my institutions and funding sources. And I'd be happy to take any questions.
Thank you, Jesse. Very interesting talk. I mean, you gave us such a clear picture of the virus evolution. And clearly, we all enjoyed your talk because there are a lot of questions. So just let's get into the discussion right away. So to start with, from what we heard, I mean, viruses mutate all the time, but with SARS-CoV-2, this virus is accumulating mutations and very smartly evolving into strain that is evading immune surveillance. And what's interesting is that these mutations seem to cost very little to the virus itself. I mean, the RBD folds properly, the ACE2 binding kinetics are not really affected. So we can just imagine more of these evolution to happen, more variants of interest and hopefully variants of concerns emerging. So does this mean that we'll have to keep our surveillance up and running, and be prepared to update our therapeutics and antibodies or even vaccine platform in anticipation of new variants, something like the flu vaccine route?
Yes, that's a good question. So I certainly think that the virus is going to keep evolving. It's possible that the rate of evolution could slightly slow down because I think right now it's unclear. I think it's probably many of these emerging lineages are partially being selected by immune pressure, but they're also partially being selected probably for enhanced transmissibility because some of them have enhanced transmissibility. And I do think that although sort of immune escape is going to constantly go on because it's essentially an arms race, the immune system is changing with the virus, transmissibility will eventually kind of be optimized. So it's possible that will slightly slow down the rate. But I certainly think that, all indications are this is not going to be something like measles where there's a vaccine and then nobody worries about it anymore. That said, I think that probably things could get much better. So in particular, we know that vaccination works quite well, particularly some of the vaccines out there work quite well in terms of reducing infection. And it will probably take a lot of viral evolution to, fully escape that immunity. And also, I think, although it's unclear, it's quite possible that the severity of disease in the presence of some level of pre-existing immunity could be greatly decreased. So even if people are reinfected with this virus every 3 to 5 years, like with a common cold coronavirus, if they get mildly or moderately sick rather than deathly ill, that would obviously, be a good thing. So I think at this point, the answer to that question is not entirely known, but I think, if we want to keep having vaccines be highly effective against infection, I definitely think they're going to have to be updated like the flu vaccine at some rate. Although we have some things working in our favor, one of them being these vaccines inherently work a lot better than any of the flu vaccines out there. And then the second one being that perhaps, we don't have to have a 100% efficacious vaccine in order to reduce disease severity a lot. But I think those remain somewhat open questions.
Interesting. And with the mRNA, the platform is amenable to changes pretty easily. So that's the other optimistic aspect about these vaccines. All right, David, you have a good question. Could you please unmute yourself and ask your question?
Sure. Hi, David Roos here from University of Pennsylvania. Thanks for the beautiful presentation, Jesse. You highlighted the running tree or ladder tree pattern of the 229E coronavirus, but my understanding is that other human coronaviruses exhibit a different kind of topology. And I was wondering whether you have any insight into what pattern we're likely to see for SARS-CoV-2.
Yeah, no, that's a great question. So I'm only going to comment on 229E and OC43, which is another common cold coronavirus, because for HKU1 and L63, they've only been discovered more recently, so it's like less historical data. But 229E has a clear ladder-like phylogeny, at least if you look at the spike. OC43 essentially has two major clades, and both of those clades have pretty ladder-like topologies within those clades, but there's two coexisting clades. And there's a nice paper by Katie Kistler and Trevor Bedford that sort of shows a lot of these trees. Why that isn't unclear. As you may even know in the case of influenza, for influenza A, perhaps this is changing for H3N2, but at least for influenza A, we've generally had a single ladder-like phylogeny, whereas influenza B at some point in the past essentially speciated into two different lineages. And, it seems like OC43 is also kind of speciated into two different spike lineages, whereas 229A has just kept one. Why exactly that is, and whether SARS-CoV-2 will sort of have a single dominant lineage or several coexisting lineages, I think is hard to predict because we don't totally understand the ecological factors that shape all of these. But I do think, it's going to be something like 229E or OC43, where there's one or a few major lineages over, a multi-year time span, rather than something like HIV where there's, essentially a continuous distribution of lineages.
Thanks. And I guess the question is also how likely that is to be influenced by the seasonality and global distribution of infection, which remains to be seen as resistance emerges.
Yeah, exactly. I agree with that. And also it's possible that in the case of SARS-CoV-2, it could to some degree be influenced by vaccination. I mean, something like the seasonal coronaviruses aren't vaccinated against, although flu is vaccinated against, the uptake is not that high and the vaccine's not that effective. So I don't think vaccination influences flu evolution that much. But for SARS-CoV-2, it's possible the vaccines will be sufficiently effective and the uptake will be high enough that vaccination will play a major role in shaping the virus's evolution.
All right.
David, did you want to ask your second question? While
yeah, I mean, I'm happy to. I don't want to monopolize the
go ahead.
So the second question is that right, David. The second question is what happens if you start with a different parental virus? We're working on experiments like that, but we don't have an answer yet. So,
I mean, and do you know whether the escape maps are similar for the entire spike protein rather than just the RBD? You showed the importance of the RBD in influencing resistance, but you can imagine, other patterns where more distant mutations could be important.
Yeah, that's certainly possible. And I mean, we're only really in a position, at least at high throughput, to look at the isolated RBD, but I think mutations to the N-terminal domain will have an effect, and possibly mutations that affect the up-down conformational dynamics of the RBD could have an effect as well. So I can actually read the questions now. Should I just go to that? Okay, so one question is, why don't we observe as much mutation in measles virus as influenza virus? Does this say something about differences in the type of vaccine or differences in the viruses themselves? So it's a really good question. And this is the kind of question that's super hard to answer experimentally. But I guess what we can say, I would say, two major theories. One, which I sort of presented near the end of the talk, is we know that the antibody response against measles virus is much better distributed across many epitopes on the virus. So that's just going to make it a lot harder for the virus to undergo phenotypic evolution, because it's, although it's not that unlikely for the virus to get a mutation that escapes at any one epitope, it's unlikely to escape at a lot of the epitopes. So that would say something about the differences in the immune response against the two different viruses, how focused versus broad that immune response is. And I don't really know what would be the basis of those differences. The other possibility is that the capacity of the proteins from these different viruses to tolerate mutations without disrupting their function might be higher in some viruses like influenza virus than in other viruses like measles virus. And I think those are probably the two dominant factors in explaining these differences, but it's hard to really decisively set up an experiment to determine which of these is the major factor or if both are the major factor. Okay, so then the next question is, a preprint came out suggesting that N501 changes the selection landscape of SARS-CoV-2. Yeah, basically, I think you're probably referring to the Sergei Kozakovsky-Pond preprint, or possibly, at least that's a good one I read recently to this. And I certainly completely agree with that. And in particular, I think that the N501Y is clearly playing a major role. I would speculate that part of what that role might be is that mutation increases the affinity of the virus for ACE2, which then might open up more evolutionary space to get other mutations, including some which might decrease affinity for ACE2. Although at this point, the virus could also be evolving to enhance ACE2 affinity overall. So yeah, I think it would be interesting to look at things in the background of N501Y, sort of related to David Roos's question. We're working on doing things like that. And then, okay, David Schatz asked, it looks like there are a couple of antibodies that had no escape mutants. How is that possible? And are these the most promising for therapy? So importantly, they had no escape mutants in that table I showed, which means that there's no mutations that are at high frequency in current viruses that escape them. They all do have mutations that escape from them. But I do think these antibodies that type of antibody is very promising. In particular, a lot of the initial antibodies that were isolated and developed for therapeutics, like, for example, the LY-CoV555 or bamlanivimab, are sort of the same sort of antibodies that infected people make, right? Because that's what people were isolating right away. And I don't think any monoclonal antibody therapeutics are being used at high enough frequency to drive viral evolution, but certainly immunity is at high enough frequency to drive viral evolution. So if I were to be developing more monoclonal antibodies, what I would do is find antibodies that bind to sort of subdominant epitopes, so epitopes that are not very dominant in the context of human sera, and develop those as therapeutics, because those are unlikely to be sites that are under a lot of pressure to change from human immunity. And so there's a good chance that there aren't going to be so many mutations arising in their epitopes. I think so far it's sort of been probably mostly a matter of luck which antibodies were chosen, whether they affected those, but I think that's something that we can do going forward. Okay, how come individual sera show different types of mutations that affect binding? Yeah, I mean, this is a good question. You know, I guess all I could really say, there's two possibilities. One is that, you know, there's some genetic or exposure differences among different people. The other one is just that the immune response has an element of randomness to it. I mean, the immune response itself is an evolutionary process in terms of making antibodies. I don't know which is the dominant answer. I do expect that these person-to-person differences will get bigger over time, because right now, everybody has had, or most people have had a pretty similar exposure history. They've been exposed to pretty much the original virus or vaccinated against the original virus. As the virus continues to evolve, different people are going to get infected or vaccinated against different variants. And so then you're going to essentially be changing the immunogen, and that's going to exacerbate these human-to-human differences in terms of the specificity. Any idea what causes some people to focus their response on one neutralizing epitope and others a different epitope? Is this just random, or do you think it's host genetics? Basically, I have no idea, but I think it's an important question. And in general, I would say immunodominance, which is the phenomenon that, you know, antibodies tend to target certain epitopes more than others, is not that thoroughly understood, and it's clearly really important. But I wouldn't say there's a deep theoretical understanding of it. Would it make sense to create an mRNA vaccine in which the spike 484 is one of several different amino acids, those that allow the most escape? It could make sense to do that, although I think probably the most likely approach that will be taken is to just make a vaccine that has K at site 484, which is the mutation that's arising the most rapidly. And interestingly, and related to the previous questions, there's two recent preprints by Alex Segal's group and Penny Moore's group from South Africa that show that people who are infected with a virus that have this E484K mutation actually make antibodies that not only neutralize that virus pretty well, but also sort of back neutralize the old E484 variant better than the E484 neutralizes forward against the K484, which I think goes to the fact that this mutation must also be changing this immunodominance hierarchy. And so I think that's, you know, an important observation that can probably be built on. So another question, is there a complex glycans at the RBD at 331 and 343? What is their effect? You know, so yeast glycosylate the RBD, but it's more mannose-rich glycosylation, so it's possible there's some effect, although so far all of the discrepancies we've observed in terms of what we see in our yeast display system and what you actually see in a mammalian cell-produced pseudovirus, we think have to do not with differences in glycosylation, but rather differences due to the fact that we're looking at isolated RBD rather than RBD in the context of the full spike, which is trimeric and is undergoing some conformational changes. So it's possible that glycosylation plays an effect, but I think the fact that the RBD is outside of the full context of spike is sort of the bigger shortcoming in our approach. So is E484K the best hand to escape immunity? So certainly E484K is the immediate mutation that's going to have the biggest effect. Now, once the virus has E484K, for most people, it's not like their neutralizing antibody immunity disappeared, it just went down. So now that E484K is fixed, those other antibodies have to target something else. And something that was previously subdominant is now going to be the new dominant thing. So there will then be another sort of best mutation for the virus. And, you know, then that process would keep repeating until there's no neutralizing antibody activity left. The complication being that then people are also potentially reinfected or revaccinated against these new variants. So then they start making new antibodies against different things. Have you looked at mutations that escape vaccine-induced plasma for a specific vaccine? Has this induced a more or less focused set of antibodies in natural infection? It's a great question. We are doing these experiments right now for one of the mRNA vaccines, and speculatively, perhaps the specificity is a little bit different, and perhaps even a little bit broader, which would be a good thing. Although I don't think I want to like hang my hat on that yet. We're still doing the experiments and looking at it, and hopefully we'll be able to put out a preprint on that in a month or so. But potentially it looks a little bit different and perhaps even a little bit better, but I can't say for sure. Why do I think the binding of RBD for ACE2 is so high compared to influenza HA for sialic acid? I mean, the reason is clearly that ACE2 is a low abundance protein. Like even on the target cells, it's not that much ACE2. Sialic acid is like everywhere on essentially everything. So flu HA uses a low affinity, high avidity interaction to attach to cells, whereas SARS-CoV-2 uses more of a high affinity and probably lower avidity interaction. Do I think that antibody effector functions other than neutralization may be important to have long-lasting immunity? Yes, probably. And in particular, I would say that probably antibody effector functions are going to be less good at providing so-called sterilizing immunity, keeping you from getting infected in the first place, which tends to require neutralization for most viruses, but they could be very important in controlling the severity of disease. And this goes to the point I was alluding to in one of the earlier big picture questions, that it's possible that, some of these escape mutants might infect people, more so than they cause serious disease. I don't think we totally know that, but there's already some work showing effector functions can be important. And yeah, somatic hypermutation, when people are re-exposed, certainly you can imagine either eliciting new antibodies against the escape variants or somatically hypermutating the old antibodies. And I think probably both will happen, although it's hard to say the relative balance. Okay, yeah. We focus on the single mutations upon analysis of multiple mutations. Have you seen anything remarkable or unpredictable? So look, I have to say I'm kind of embarrassed by this because my lab supposedly studies protein evolution and epistasis. And in fact, Tyler Starr, who's led a lot of this work, his whole PhD was on studying epistasis. We wanted to look at those questions more, but we started working on this topic where people cared so much about the effects of single mutations. We've never made it past the single mutations in any meaningful way, but we're now getting there. So at this point, I can't really draw out any conclusions, but I think that hopefully we'll be able to find stuff there. Tiago Graf asks, are you planning to test how indels affect immune escape? I think this is certainly important, particularly in the N-terminal domain of the virus. There's a lot more indels, especially dels, than point substitutions. And we're working on trying to make an NTD type library, and we're trying to put some indels into that, although we don't have a comprehensive way to make all the indels, sort of like we can all the point mutations. On one of the immunity preprints, B.1.351 neutralized wild type, and wild type did not neutralize B.1.351. This is sort of exactly what I was referring to in one of the earlier questions. And yeah, as they point out, there was a particular person that was infected with E484K. I believe this is the Alex Segal preprint that had amazing neutralization of both wild type and B.1.351. I think all we can say, and this is what they basically say in that preprint, is it's an N of 1, right? There's one person in there who is infected with just the E484K virus, and they do have this kind of amazing neutralization. But with that N of 1, I don't think it's possible to say whether it was just that person made a really good antibody response, because these are variable across people, or if there's something really good about that wild type plus E484K virus. But this is certainly an important thing to look into. Okay, so the next question is there a reason why the polyclonal immune response to influenza is or has evolved to be so focused? Again, I don't really think we know the answer to that exactly. I mean, it's unclear, at least to me, it's unclear exactly what determines what's an immunodominant epitope for a polyclonal antibody response. But I do think it's an important thing to try to figure out, but I don't have an answer really. All right, how do you think about antibody versus vaccine escape, especially the strong 484 position? So I think what the fact that 484 certainly escapes a decent number of monoclonal antibodies, but by no means all of them, because monoclonal antibodies bind all over, but it has such a big effect for polyclonal serum, indicates that the particular class of antibodies that bind to the epitope that include 484 are dominant in serum. So either there must be more of those antibodies relative to the other ones, or those antibodies must have more neutralizing activity relative to the other ones, or some combination. And so that's why, even though that's only an escape mutation for certain classes of antibodies, it's so important for serum. When testing viral fitness, why would we care if the RBD folds correctly or not? As long as there is high affinity for ACE2, wouldn't we conclude that's proof of the pudding? I mean, yes, basically. So I agree that if it binds to ACE2, that's good enough. But I mean, in practice, it's not going to bind to ACE2 unless it folds correctly, because it's a very specific interaction. And I think at a conceptual level, distinguishing between the two ways that you can alter ACE2 affinity, one of which is just to misfold the entire protein, and the other one, which is to specifically perturb the interactions between the RBD and ACE2, is a useful distinction to make. But I completely agree, if the RBD is misfolded, it's not going to bind to ACE2.
Yeah, wow.
Okay,
well,
I think I made it through all those questions.
Yeah.
So I don't see anything more in the chat anyway. If I missed something.
Audience, thank you for asking such amazing questions. And Jesse, thanks for making Sarada and me irrelevant. That was remarkably efficient. I have a tiny follow-up on the question of testing for epistasis in the presence of, other beneficial mutations. Just, the other side of the coin, you showed us, single nucleotide mutations. Are we getting anything like silent mutations that are fixing, but then would open up
certainly
additional steps?
There are some number of synonymous mutations that have fixed in the virus, probably including the spike. Honestly, I haven't paid much attention, so I can't tell you what they are. And you could certainly imagine that in some cases, those synonymous mutations will change which amino acid mutations are then most readily accessible by subsequent nucleotide mutations. And so certainly, I think, particularly if you wanted to forward model the evolution, that would be important to look at. I haven't looked at it all, though.
Yeah. And then, we know we have to go in a couple minutes, but maybe just one more. I guess our useful audience is. Okay. Just someone was thanking you. Yeah. Various people like Jason McClellan have been in the news saying like SARS-CoV-2 is out of tricks. Because you would get convergent evolution of E84K, Del 6970, etc. And the fact that these are showing up so much, people are saying, hey, maybe it's already near peak fitness.
So first of all, I'd like to defend Jason McClellan for being at the news, because now, having had to give a fair number of these interviews myself, I've learned that it's like super easy to say like one thing, which then taken out of context sounds overly simplistic, because Jason McClellan is clearly like one of the world's experts on this and understands what's going on really well. But I do think, to Jason McClellan's point, it is possible that some of the easiest hit mutations have already arisen, like E484K, but I don't think the virus is going to, there's a lot of sites that are tolerant of mutations, and I think the virus's antigenic evolution is going to keep going. Like I said, I think there's a couple of reasons that the rate could potentially slow down. One of which is that some of what we're seeing right now might be co-selection, selection for enhanced transmissibility, which is also changing antigenicity. But I do think that the virus is not going to, stop antigenically evolving next year or something. I think this is going to be a continuing process. And we can see this with coronavirus 229E and, sort of any other antigenically variable virus.
Yeah. And of course, it's going to be under much greater selective pressure for antigenic escape as we all get vaccinated, hopefully. So.
Yeah, probably. Although, there may also be then fewer total infections. So I think it's a little bit hard to predict exactly how that's going to alter the rate of antigenic evolution.
Yeah. Jesse, mind-blowing. Thanks as always.
Okay, thank you.
Thank you, Jesse.
So Matt, can you toss up the slide? So important, there will be no talk next week. So this is in 2 weeks. Come join us to see Iran Segal talk about the course of the disease and vaccination campaigns in Iran and other places. So have a nice week off, and everyone be safe, and we'll see you in 2 weeks. Bye-bye.
Thank you, Jesse. Thanks, the audience.